Rapamycin derivative longevity script. There's a worm right now living in a lab in China that is genuinely older than it has any right to be. By worm standards, it's ancient, practically geriatric. And the thing that did it wasn't some expensive genetic therapy or a futuristic procedure. It was a tweak to a drug that's been around for decades, a drug originally isolated from bacteria found in Easter Island soil. But here's the question that should keep you watching. If we can make a simple chemical modification to extend life in one organism, why hasn't this become standard practice in everything from mice to humans? Why are we still not doing this everywhere? The search for the fountain of youth is, well, basically as old as humanity. But it's only been in the last couple of decades that we've discovered something genuinely startling. Some drugs actually work. Not in the sense of unproven supplement bottles sold by podcasters, but in the rigorous, testable, repeatable sense. Rapamycin is one of those drugs. Scientists discovered in the early 2000s that this immunosuppressant, a drug originally used to prevent organ rejection after transplants, could extend lifespan in mice. That finding cracked open a door. If a drug designed for one purpose could accidentally do this, what if we engineered drugs specifically for it? But rapamycin had a problem. It suppresses the immune system, which is great if your body is attacking a transplanted kidney, but terrible if you're trying to live a long and healthy life. You'd be trading years for vulnerability. So researchers started asking, can we get the longevity benefit without the immunosuppressive downside? This study is an answer to that question. The work was done by a team at the Fujian Institute of Microbiology in Fuzhou, China, published in the Journal of Applied Toxicology in April 2026. The lead researchers were Li K, Li Q, and Jie L, working alongside a handful of collaborators. The team was housed in the Fujian Provincial Key Laboratory of Screening for Novel Microbial Products, which sounds like exactly the kind of place where you'd design new drugs. This wasn't some fringe operation. It was published in a legitimate peer-reviewed journal, the kind where the data gets scrutinized by other scientists before it sees the light of day. So what did they actually do? The researchers took rapamycin and modified its chemical structure. Think of it like this. Rapamycin is a multi-tool that does several things at once. It's antifungal, anti-tumor, anti-proliferative, and immunosuppressive. The problem is, it's doing all those things simultaneously, and some of those effects are unwanted if your goal is just to live longer. So they created something called FIM-X8, a rapamycin derivative, basically a chemical cousin, that kept the longevity benefits while dampening the immunosuppressive side effects. Then they tested it on Caenorhabditis elegans, which is the standard model organism for aging research. These are tiny roundworms about 1 millimeter long, transparent, with exactly 302 neurons. They live for about 2 to 3 weeks under normal conditions. This makes them perfect for aging studies because you can run a complete lifespan experiment in weeks rather than years. The researchers dosed the worms with FIM-X8 and watched what happened. They measured how long the worms lived, how they handled heat stress, how they responded to oxidative damage, the kind of cellular damage from free radicals that accumulates as you age. They also looked at which genes were activated when the worms got the drug, diving deep into the genetic mechanisms of what was happening. So what actually happened? The worms got older, significantly older. The lifespan extension was substantial enough to be unmistakable, but here's where it gets interesting. When the researchers looked at which genes were doing the heavy lifting, they found something specific. A gene called RSKS1 appeared to be crucial. When they used mutant worms that lacked RSKS1, the longevity benefit largely disappeared. This is important because it suggests the drug isn't working through some vague, magical process. It's working through a specific genetic pathway. They also saw that the worms handled stress better. When exposed to high temperatures or oxidative stress, the treated worms survived better than untreated controls. The mechanism involved the mTOR pathway, which is a master switch in your cells that regulates growth and metabolism. Think of mTOR like the cruise control on your car. Normally, It's set at highway speed. Your cells are growing, reproducing, metabolizing actively. Rapamycin taps the brakes, slowing that system down. That slowdown apparently extends lifespan, at least in worms. And FMX8 does this without triggering the immunosuppression that makes rapamycin problematic for long-term use. So what does this actually mean? Well, first, the honest caveat: this is worms. Worms aren't humans. Your immune system is incomparably more complex than a nematode's. Your metabolism is different. Your lifespans are measured in decades, not weeks. So while this is genuinely interesting, it's not yet a green light to start taking modified rapamycin tomorrow. But it's a signpost. It suggests that the strategy, finding drugs that trigger longevity pathways without the downsides, is workable. In practical terms, if this could eventually translate to humans, we're talking about a drug that might extend healthy lifespan without making you immunocompromised. That's the dream. That's what researchers have been chasing. The fact that they found a chemical modification that achieves it in a model organism means the approach itself isn't flawed. It means it's just a matter of iteration, refinement, and testing. The limitations are real, though. One major one: we don't know if the effect would hold at doses safe for humans. In worms, you can push concentrations quite high. In humans, you'd need to navigate tolerability. Another limitation: we don't know if lifespan extension in worms translates to healthspan, the years where you're actually healthy and functional, in larger organisms. What's the point of living 10 years longer if you're frail the whole time? Where does this fit in the larger aging research landscape? Rapamycin derivatives are becoming a crowded field. There are dozens of labs working on versions of this exact strategy. Take the longevity signal, preserve it, dump the side effects. Some are using rapamycin itself, but at lower doses. Some are creating new compounds entirely. The basic science is solid. mTOR inhibition really does seem to extend lifespan across multiple organisms, but we're still in the phase of optimizing the tool, not deploying it clinically in humans. That's probably still years away, depending on funding and regulatory appetite. The bigger question this raises is structural. Why do drugs designed for one purpose, immunosuppression in transplant recipients, accidentally extend lifespan? It's because longevity isn't some mysterious force. It's the outcome of biochemical processes. Find the right lever, pull it, and things happen. FMX8 is a slightly better lever than the original. Eventually we might find an even better one, or even a whole different lever in a different pathway. That's the optimistic future. The pessimistic one is that aging is so multifactorial that no single drug, no matter how well engineered, will give us the dramatic extensions we're hoping for. So here's the single most arresting thing about this study. It suggests that immortality, or at least substantially extended healthy life, might not require some impossible sci-fi technology. It might just require better chemistry. Scientists have taken something that nature made, tweaked it slightly, and the result is organisms living substantially longer. That's not theoretical. That's done. The question isn't whether it's possible anymore. The question is whether we can do it in you, and whether we have the stomach to actually try. Turns out, scientists have been busy, and they're not done yet.